Thank you to the organizers of Thirsting for Theoretical Biology and the three V's who are the organizers. It's very nice to be here. I'm going to tell you a little bit about the work I've been doing for approximately the past 7 to 8 years on a particular question that involves both active matter as well as the understanding of the cell nucleus and ways to combine both of these together. So if it may help a little bit to tell you what my interests are, I started off as a physicist. So there's a before and a now. My before involved statistical physics, soft matter physics, and a bit of biophysics. Now it's very largely biophysics, computational biology, but at least over the last year or so, thinking about diseases, in particular COVID-19, has pretty much taken over my entire research agenda. And the shift happened approximately 10 years ago, around 2010, when I moved from thinking about more physics problems to thinking about more biology problems with a physics flavor. My real motivation coming from the background of a physicist has been to think about this question of, can you bring to biological modeling the rigor of models in physics? And I want to sort of pose this question in a very specific way by showing you this picture. So for the biologists in the audience, it doesn't really matter what this is. This is some fit of some experimental data. The experimental data are the little squares that you can see. The fit is the line that goes through these squares. And the experimental data is, of course, background cosmic microwave spectra as obtained from an experiment called the Kobe experiment. The line is a fit of a particular curve, and that equation to that line is what's given at the bottom, the 2hc squared by lambda 5,4, and an exponential function to the right-hand side of that. And you can see that it does a perfect job of fitting the experimental data. In fact, there are many things that are unusual about this particular graph. This is a graph that looks at electromagnetic waves that come in right from the furthest reaches of the universe and are measured on Earth. But even given that, the error bars on each of those points, each of those little squares, are actually about 100 times smaller than the size of the square itself. So that points to the extreme accuracy that experiments in physics are capable of. And the fit between the theory and the experiment is also so precise that the only real argument happens if you look at the extreme ends of these two and to see, are there little wiggles there? Do they sort of have meaning or do they not have meaning? And those wiggles are so small that you really can't see it on this particular plot. So that's an example of the success of the nature at which you can compare experiments in physics to theory in physics. If you thought about the same thing in biology, if you looked at the nature of experiment in biology and you ask the same question, can you bring to biological modeling the rigor and precision of models in physics, it's clear that biological data itself has it sort of comes with an irreducible intrinsic noise component. So just look at the picture on the right-hand side. I've cropped the axes because what it is doesn't matter. It's just some spread of experimental data from which a particular inference is drawn. You can see that there is a very substantial spread in the data. In fact, if those top 2 points were not there, you would be hard put to say that there was any real difference between these data at all. So biological systems are intrinsically noisy. And for the systems that I consider, they're also small, they're soft, deformable, confined, crowded, and out of equilibrium. And all of this applies equally well to the previous talk by Anjana, that she's looking at things confined to the scale of a bacterium, which is small. She's looking at DNA, which is a soft, deformable molecule. It's confined to a small region here. It's extremely crowded with all sorts of other types of molecules of different shapes and sizes. And some part of her work is, of course, related to the fact that there are out of equilibrium processes that happen in the background of this, ATP-consuming processes that happen in the back of this. So having made this distinction between sort of the relative cleanliness of experimental measurement in physics and the fact that you do have very accurate theories that deal with that can model that to extreme accuracy. And then we contrasted that to biological data, which is telling a story, but it's hard to figure out what the essentials of that story are that survive the sort of statistical noise and all of these other components that I built into it. So then this raises a bunch of questions. What would you call progress in the quantitative modeling and understanding of specific systems in biology? What is your benchmark? What is your sort of minimum program for saying that a theory works or it doesn't work? Then the question is, what are the questions we could ask? And together with that is a sort of what sort of answers would satisfy you as potential answers to that question. And one needs to understand this in advance in order to not set one's sights too high in terms of what you can deliver versus the specific problem that you want to think about. Again, because the word model often confuses people who are not used to dealing with this. It happens, in climate models, any other type of model, or is evolution, it's just a model, or is it reality? All of these confusions exist. Remember that whenever we talk about a hypothesis, what we're doing is really constructing a model out of words. And what a physicist or a mathematician or a statistician does is to be able to look at these word models in a quantitative manner. And a model is something that leaves out a lot of detail, but yet attempts to capture features that are essential. And this is the ultimate goal of figuring out what is an essential description of whatever it is that you're looking at. Anjana's told you about very specific details underlying dsDNA repair in bacteria. But then the question is, of all of the biological detail that we know goes into that particular problem, into the specification of that problem and the results that came from it. If we were to think about it from a modeling perspective, what is it that we would absolutely need to include? What is it that we could include but is not that important? And what is it that's unimportant to include at the level of obtaining a deeper understanding? So we should understand where we should set our sights when we think about the role of models in theory. So when I said leave out detail, capture essential features is the ultimate goal, I guess most of the speakers have pretty much said these things, people who work in modeling. But always the question is, what details do you leave out? And the problem that really is behind all of this for people who think about quantitative descriptions of biological systems is there is no well-defined, universally acceptable way of doing that in a general context. People may sort of fool you about this in different ways, but this is true, that very much what you leave in and what you take out is very much a matter of the individual modeler's own ideas behind what is important. And at best, what helps you do this is to use intuition that you can get from similar studies. Plus, of course, the overarching knowledge that physics and mathematics, in quotes, work. They are very successful descriptions of reality. So after having completed that somewhat general introduction to models and to thinking about models, let me come to the question that I've been interested in and try to illustrate some of those ideas and some of that background in terms of a specific system of interest. And the question here that's been motivating us is what underlies large-scale nuclear architecture in metazoans? And how does one think about this as a problem at the interface of biology and physics, where one cannot address this purely in terms of biology. One has to have a physical understanding of what's going on. And as I said, the real aim is to have a quantitative understanding. So what is the quantitative information that you can derive from a theoretical model that you can feed back into your understanding of what determines biological features of large-scale nuclear architecture? Metazoans are multicellular eukaryotic organisms that form the biological kingdom Animalia, animals. So we exclude plants with their rigid cell wall from this. Here's a cartoon, it's a typical picture of a single cell. What we're interested in is material inside the nucleus. There's lots of stuff outside there which other speakers have spoken about. For example, the properties of the plasma membrane that encloses the cell, the properties of the nuclear membrane that encloses the nucleus. And we won't be talking about that. We'll only be looking at the nucleus and the content of the nucleus. And of course, what happens inside is background to other types of biological processes that are in eukaryotic systems compartmentalized. There is transcription that happens within the nucleus, and there is translation that happens within the cytoplasm, where the DNA is converted to messenger RNA, then later into protein. So this is important. Again, here we will not be concerned with processes that happen that are cytoplasmic processes, but only with what happens in the nucleus, and specifically processes associated with the transcription and the consequences of that for how the nucleus is structured inside. Again, general background, typical cell, excluding sex cells, there are 23 pairs of chromosomes in the nucleus. Each chromosome is a single long DNA molecule. And for a sense of scale, if I take all the chromosomes inside a typical human cell, lay them out end to end, tie them together at their ends, I will have a linear structure that is about 2 meters long. If I multiply that 2-meter number by the 10 trillion odd cells in the human body, that gives me a total length in meters of something that is enough to go from the Earth to the Sun, approximately about somewhere between 80 and 100 times. So that's a lot of molecule, there are lots of linear molecule that is contained inside a cell. It's all packaged into a very, very small space of order of about 10 microns inside the nucleus. And the fact that we have to deal with long DNA molecules implies that to think about the polymer aspect, the polymer physics of these molecules must be important or relevant to understanding how the nucleus is structured, because these are our essential components of what's inside the nucleus. So 23 pairs of chromosomes, each a long polymer. Question is, how are they distributed? And one way of thinking about it is that is distributed according to that picture that you see here. You see these molecules, they're long, they're extended. This is a cartoon picture, a sort of a noodle analogy of what might be happening inside the nucleus. And then the question is it just a noodly mix, something that looks like this? Or is there any more structure that is superimposed on top of this? Again, what makes a simple polymer or a set of simple polymers or 23 pairs of simple polymers different from DNA in its natural milieu in the nucleus of a living cell? So the first thing to note is that it's not just bare DNA a la Watson and Crick. What we have to remember is that DNA is packaged at multiple levels inside the nucleus. It's a DNA, the level is base pairs bound into a double helix, but then the level of packaging that sits at one level above is the histone octamer, 8 histone proteins that in pairs are tied together, forming a little round object, because that is DNA in solution is weakly negatively charged, whereas the histone proteins are positively charged. This introduces an electrostatic interaction between them that leads to the DNA winding around the histone protein. And then these combinations of histone protein, the DNA plus histone protein is called a nucleosome. Nucleosomes are further hierarchically organized into these spiral helical type of structures that are even more tightly packed at further levels into what we think of as a chromosome that we see at the point at which the cell divides. Okay, so DNA plus histones plus accessory RNA and proteins that bind to DNA, all of this together is the natural milieu, the natural background in which you see DNA within the nucleus, and this is called chromatin. So now we've gone from talking about the sort of level that Anjana was describing, where you had DNA and DNA repair proteins associated with that. We're now looking at eukaryotic cells, not bacteria as she was, but we're thinking about questions of what is the complexity that defines DNA within the nucleus. And at some level, it's a complexity associated with packaging a long molecule into a small region. And that packaging is accomplished very cleverly by using electrostatics to take negatively charged and positive charges and put them together, ensuring that they come together, as well as many other more complicated versions of many levels, at least 3 or 4 different levels of hierarchy in the nature of this packaging until you reach something that is very compact. So the word chromatin is important here, and we'll now be talking about chromatin in terms of the stuff that attaches to DNA and conducts various types of non-equilibrium processes on DNA. And then we want to ask, how is that relevant to the structure of chromatin, the structure of chromosomes within the nucleus? So the word chromatin, to put a little bit of history back in, goes back a long way. It was coined in 1882 by Walther Flemming to designate that substance in the nucleus which, upon treatment with dyes, known as nuclear stains, does absorb the dye. So chroma, meaning color, chromatin is where it's because it is stained by dyes designed to dye nuclear material. So the stainable substance of the nucleus is what he called it initially. That's what the old pictures look like. We need to talk about chromatin when we discuss the polymer physics of chromosomes inside the nucleus of living cells. So that's what, as I said, that's again the cartoon picture of how you go from the helix to compacted DNA to the large-scale chromosome structure. So let me tell you a little bit about how our thinking about chromatin has evolved, at least certainly over the last 30 to 40 years, but with a lot of input from basically 1880 onwards, 1890 onwards, when people first began to understand that the nucleus was a more complicated object and had a certain definite substructure inside it. So here's a quote from a nice review article by Kremer and Kremer, who says, during the 1970s and the 1980s, most researchers seemed content with the assumption that the nucleus is filled with intermingling chromatin fibers and loops like a dish of spaghetti. So that's a dish of spaghetti to the left, the old picture that I showed you. So that's what we used to think about chromatin. And of course, things have changed since then. What we do know now is a far more complex and nuanced picture, that chromosomes are territorial in character. They're not all mixed up like this pasta that you see on the left-hand side, like the spaghetti. And what is more, these arrangements of chromosomes are not random. It's not as though any chromosome can be anywhere. If you look at a particular type of cell, a particular cell type, there is a certain preference for a chromosome to be more interior versus more exterior. So that's the picture that you see in that inset slide, where you see chromosomes differently colored. There's a yellow one, a blue one, a green one, etc. You see the nucleolus in there. And you can see that far from being all intermingled, as in that spaghetti analogy, they tend to sort of have their own little spaces carved out for themselves. These are called chromosome territories. And this is the root of why I said chromosomes are territorial in character. For all of this talk, I'll be talking about human cells in interphase, which is the period in between cell division, because interphase occupies about 80% of the cell's life between division. And that's the point at which you don't see the separate entity of the sort of the H-shaped chromosomes that I showed you in the picture, which you see at the point of division at mitosis. But over here, it's much more diffuse and spread around. And this is of some interest to us, that even in this diffuse spread around state where you don't see the chromosome structure explicitly, why is it that a territorial structure is preserved? And what determines the non-random arrangement of these territories? So not mixed at random, but found in well-defined territories. And that's going to be our sort of one point at which we'll have to understand why this is so. If I took an ordinary polymer, shook it about in 23 pairs of polymers, shook them about, would they be all mixed up? Would they be unmixed? And what determines this? How do we know what we know in this particular case? And here, the fact that chromosomes form territories is obtained through a technique called chromosome painting, or the fluorescence in situ hybridization method, where you attach fluorescent tags to specific sequences of DNA. And then you can have sequences that are peculiar to the chromosome 11, chromosome 8, chromosome 1, chromosome 4, etc. So you can ensure that chromosome 11 is more or less painted yellow, chromosome 2 is painted more or less blue, etc. And then wind up with a picture that looks like the picture below, where you've assigned a chromosome to each of those colors that you see. And the fact that you have this patchy structure with the colors somewhat separate is indicative of the fact that you have territoriality. The observation of what is called heterochromatin and euchromatin is a much older observation, and that comes from the fact that it goes back to Emil Heitz in 1929, who suggested that chromatin, which we understood earlier, was divided into somewhat more condensed, less active regions, which are largely devoid of genes. He called that heterochromatin, and separate from gene-rich domains, which he termed euchromatin. So tightly packed DNA, gene-poor heterochromatin, tends to sit a little more on the outside of the nucleus than loosely packed DNA, which is gene-rich or euchromatin. Okay, so that's one more sort of region. So we've learned about territoriality of chromosomes. We've also discovered that in terms of packing, there is a distinction between more tightly packed and less tightly packed. This is a distinction that is seen in EM. And this also corresponds to whether that the sequences of DNA in those tightly packed or weakly packed regions are gene-rich or gene-poor. So now we can ask a little more focused question. We started thinking about DNA as polymer, as a simple polymer, as a long molecule. Then we said it can't just be a long molecule. There's lots of stuff attached to it that's needed to compact it. Then we said, okay, let's look at this more compact object inside the nucleus, call it chromatin. Let's ask, are there any peculiarities with how chromatin is packaged inside the nucleus? And it turns out there are lots of peculiarities. There is a distinction between heterochromatin and euchromatin in terms of packaging. And there's also a distinction at the level of individual chromosomes. They tend to prefer certain regions of space relative to the nucleus center, and they tend to form territories. So the formation of chromosome territories, the location of chromosome territories, and the distinction between euchromatin and euchromatin, the spatial separation between euchromatin and heterochromatin, is going to be central to our sort of investigation of why this might be so from some sort of biophysical model. So to do that, we again need to ask this question: what makes a simple polymer different from DNA in the nucleus of a living cell? So the biological context of this is that we are not allowed to do the one thing that is most natural for us to do. That is to think as though that we are in thermal equilibrium. Chromatin is not in thermal equilibrium. And that's the single most important point behind the modeling that I will describe to you. What does it mean? So I imagine that people have spoken about this earlier, about what it means to be out of thermal equilibrium. But one example of something that is not in thermal equilibrium is the relationship between the Earth and the sun. The Earth receives energy from the sun and it sends it out. So there's constantly a flow of current that passes through. There must be as much coming in as going out, because otherwise the Earth would just either heat up or just cool down. So balance is important. So the net flow of current in steady state is very much characteristic of a system in thermal equilibrium. Out of thermal equilibrium, which means that it is not in thermal equilibrium. And so you can think of this example as one sort of motivating example for the ideas that we're going to think about. But at a more microscopic scale, which is really the context that we want to look at, systems are driven out of equilibrium because they consume a molecule of ATP and break it down into ADP. So the ATP hydrolysis releases energy. The ratio of ATP to ADP is maintained out of thermal equilibrium. This releases energy on the microscopic scale of a few nanometers. So you have a molecular motor there beginning to step. And this really is how all these cells function, the basic energy conversion mechanism of all of these cells. And the fact that this is so generic suggests that living systems are out of equilibrium in a very specific way. And this is what we now refer to as activity. Okay, so now you can see the cycle between ATP hydrolysis to form ATP, the release of the PI, and so on, as you can see from this particular picture. This is a completely energy-driven process. And the fact that it's out of equilibrium is really responsible for the fact that the motor can move in one particular direction at a fairly steady pace. So I said energy input at the nanoscale in biological systems coming from ATP hydrolysis. This is called activity. And we should really be thinking about chromatin as an active polymeric system. So that's where now we have managed to push the question to. We started off thinking about polymers, went to chromatin, and then finally discovered that it had to be even more complicated than that. To actually be able to mimic what is happening inside the nucleus, we need to expand our notion of polymers in equilibrium to polymers out of equilibrium, and to think about these dense mixture of polymers inside the nucleus as some sort of very special non-equilibrium phenomenon that we need to try and understand how to quantify.
Gautam,
Yep.
is it possible that you can take a few questions now?
Happy to take questions, yeah.
Okay. Debashish, do you want to unmute yourself and ask the question?
Yes, thanks. So, Gautam, I had a question. This localization of heterochromatin towards the nuclear surface and euchromatin towards the middle of the nucleus. So, is this independent of cell type, or this can reverse in different cell types?
This is largely independent of cell type, but there are specific counterexamples. And I think there's sort of, there are, I think, some mice that go out at night, for which is completely the reverse. And you have the euchromatin towards the outside and the heterochromatin as a central component. But by and large, the sort of more peripheral heterochromatin is the norm.
Okay.
In biology, there are always exceptions. So we're going to sort of talk about what is more or less the norm.
But for example, like different cell types of specific species,
yeah.
But let's say in human, that doesn't change from tissue type to tissue type.
There are some variations, and I'll come to that as we go along.
Okay. Okay.
Vidya, would you like to ask your question?
Yeah. Hi, Gautam.
Hi.
Is there any distinction in these patterns of packaging between facultative and constitutive heterochromatin?
This is not something that we looked at, and there is certainly a distinction. And as you know, you can sort of change as you make it more active. You can change the locations of heterochromatin regions as they move inward. This is usually accompanied by some change in position. That if you sort of activate heterochromatin that is peripheral, you tend to move it a little more interior, where it becomes more euchromatin-like. So these are the issues that I'm going to be concerned with, but the modeling certainly doesn't make this distinction.
Thanks.
Pratyush, do you want to ask your question?
Yes. Hello, sir. I wanted to ask that you mentioned how living systems are not in equilibrium. But then you gave an example of the Earth and Sun energy dynamic. But I was wondering if you could actually explain this for once more and tell us how are living systems not in equilibrium? Because when we look at even other systems in thermodynamics or physics, we almost always assume that something, let us assume that this is not changing, and then we call that system at equilibrium. In real world, finally, there are no really systems at equilibrium. So why can't we look at living systems as systems that we're just not looking at with the right assumptions?
So that's actually a very good question. So there are parts of living systems that are in equilibrium, that are completely consistent with the standard rules of how you do biochemistry and how you find rate constants and so on and so forth, because by looking at the ratio of products to reactants. So there are parts of living systems that are in equilibrium, and there are parts of the living system that are not in equilibrium. And typically those parts have to do with the consumption of ATP, and the ratio of ATP to ADP is held out of equilibrium because ATP is constantly being made. Your body turns over roughly about half of its body weight in ATP every day. And it's important to maintain this distinction and to ask, if I assume that it was at equilibrium, would these following phenomena happen? And the answer is, you would never get a molecular motor to move in one direction if it were in equilibrium. The fact that you can do work in a targeted manner, that you can move unidirectionally, or you rotate in one direction, all of these require at its very core that you have to be out of equilibrium. This is something that we've really understood only over the last 20 to 30 years in a very, very targeted sense. So the best way of saying it is that it's not a very general statement. There are parts of biological systems typically sort of modeled using system biology methods with standard biochemical rates that are perfectly well described by ideas of equilibrium. But there are other parts that cannot be, and typically these have to do with some sort of mechanical components that need energy to drive them. And these mechanical components must be driven by a non-equilibrium, an out of balance concentration of ATP to ADP.
Okay, thank you. Thank you.
Last question for now from Sonali.
Thank
Sonali.
you, Professor Gautam. Yes, I have a question. Does the observation of chromosomes in non-random territories imply that organization encodes information?
I'll come to that a little later, and that is among the things that I will discuss. It's certainly true that we believe that they encode information. What that information is, we don't quite know. I hope to clarify that as I go along.
Yes, Gautam, you should go along now. Thank you.
Okay, so here are the main points that I've made so far. That chromosomes are territorial. Euchromatin typically tends to be more central, heterochromatin more peripheral. That individual chromosomes are often radially positioned by gene density, sometimes by size. I didn't show you the data that corresponded to this. But then there's a whole lot of other things that have attracted some amount of attention over the past 30, 40 years. The inactive and the active X chromosomes typically tend to be differentially positioned. Active alleles tend to be closer to the nucleus center than inactive alleles. There are various regularities in chromosome shapes and structures and roughness and ellipticity, etc., that need explanation, but just have not been discussed so far. So here, it's a whole bunch of, you can call them observations, very generic observations about behavior across multiple cell types that seem to share these common features. And as I said, biology is full of exceptions. So much of what I will say here really corresponds to the average type of behavior, but you can always concoct an exception in which the relationship between euchromatin and heterochromatin is opposite. And there may be specific biological reasons for that, which we don't understand as yet. But let me go along and then I will explain some of these terms as we go. So first of all, when I said non-random, what do I mean by non-random and how do I quantify it? So the idea here is if I think of the nucleus as a sphere, and I draw little subspheres inside it, little shells inside it, and I can look at, for example, all of the DNA that corresponds to a particular chromosome and ask what fraction of that falls within a particular subshell, as I've shown here. So look at the picture on the left where you have a sort of little more dense towards the inside, a little more dilute towards the outside. So if I asked what is the relative amount of DNA that I might find in this particular case, it's just these little circles that you can see. As I move outward radially from the center, you can see that it sort of goes up and then it comes down again. And that shape, that characteristic shape, is some indication of the distribution of these circles inside there. And I want to extend these circles here in this particular graph and this particular schematic to thinking about the center of mass of particular chromosome or the distribution of DNA associated with particular chromosomes. Look at the picture on the right where you can see that it's depleted in the center and tends to be more, there are more circles on the outside than towards the inside. So this would have a distribution that peaks more towards the outside. It's sort of small here, goes up and then comes down. And you can see these are very different distributions. And this is one way of quantifying what you might call a non-random arrangement. So the challenge would be to predict this. Take a whole bunch of nuclei, look at the distributions, make a graphical representation that you can see there of where the DNA associated with each chromosome is, convert that to a statistical representation by averaging over all of these data. And as you move from the center to the periphery, you plot the same sort of histogram that I showed you. And the observed frequency is the fraction of time that you will see a little piece of DNA of chromosome 1 or 13 or 15 in a certain box at a particular radial position. And the shape of this curve is going to be something that experiment can dictate to us, but is a challenge to theory to predict. So there's a term that is used in some of the economics literature called a stylized fact. I didn't like that term too much initially, but I think now I feel a little better towards it. So these are facts, experimental facts that come from experimental observations, but nevertheless they're stylized in the sense we sort of assume that these are general features that we need to try and understand. The peripheral gene poor chromatin, interior gene rich chromatin that we talked about in the heterochromatin, euchromatin context. The fact that chromosomes form territories. The fact that chromosome positioning is by gene density is the plot that you see on the bottom left. And there you can see this is the distribution of DNA for two chromosomes, chromosome 18 and chromosome 19. Chromosome 19 is a darker color. You can see it peak more at a more interior point and then go down. So between 0 and 100 is the radius of the nucleus. So relative radius starts at 0 at the center of the nucleus and 100 is the periphery. And you can see the distribution of chromosome 18 and the distribution of chromosome 19 is very different. So that's certainly something to understand. It looks as though the more gene-dense chromosome, which is chromosome 19, which contains a lot of housekeeping genes, tends to be more towards the inside, just as in the picture that I showed you earlier, whereas chromosome 18 tends to be more towards the outside. The picture to the bottom right is an example of chromosome positioning by size. So on the x-axis is the size of the chromosome. So 1 and 2 are large chromosomes, 21 and 22 are small chromosomes. So the smaller chromosomes tend to be a little more towards the inside. The y-axis is the relative radial position. Small radial position towards the center, large radial position towards the outside. You can see this sort of gentle slope in the data, which suggests that maybe chromosomes are being positioned by size in some of these cases, if you look at all of these chromosomes. And now the question is, what's right? And where, given data like this, what is it that you can actually say? Here's how one would quantify a graph that you see over there. If I said the probability of finding a little piece of chromosome at distance r, at radial distance r from the nucleus is p of r, I multiply that by 4 pi r squared to give me the size of the disk at distance r, and I call this function S of r. If p of r was completely constant, if it didn't change at all with location r, then S of r would just be the 4 pi r squared multiplied by some number, which would just go up. So it would be small near the center of the nucleus and large towards the periphery. This is what you would expect if there was an equal probability of finding a chromosome anywhere inside the nucleus. But what you actually see is the picture on the top left. You can see that it has far more structure. The one that is less gene dense is the one that seems to resemble the radial symmetry case on the right-hand side a little more. In the center, its peak is more shifted outward. But chromosome number 19, which is gene dense, is the one that is shifted much more towards the center. Okay, so this is sort of the comparison between what one might have expected from a theoretical point of view, assuming that chromosomes were free to move around anywhere at random, and what one sees in the experiments. So let's go from the physical context, which is energy input happens at the nanoscale. This corresponds to non-equilibrium. We'll call it activity. And now we'll come to a hypothesis. And the hypothesis is that it's activity, which is inhomogeneous across gene-rich and gene-poor regions, which determines all of these features. By all of these features, I mean all of these features, these stylized facts that I listed, the formation of chromosome territories, the preferential location of one chromosome versus another, the peripheral versus interior chromatin, etc., chromosome positioning by size. Let me just take a quick break to answer the door, and I'll be back in exactly 30 seconds. Apologies, that was a loud doorbell that I predicted would happen sometime in the middle of the talk. Can I go on, Sandhya?
Yes, please do.
Okay, so that's our hypothesis. There is activity that is responsible for all of this interesting behavior. And now we want to try to quantify that. First of all, where does activity come from in this context? The answer is that when you take chromatin in living cells, there are many, many energy consuming processes that act upon it. And all of these processes typically will involve putting some force on some local part of the chromatin. There's a whole bunch of chromatin remodeling that goes on. There are about 60 different enzymes, the switch, SNF, the RSC, the I-switch, etc., that basically open up and close little regions of chromatin, depending upon the signals that are contained in the histones that are attached to the chromatin. The act of transcription itself is a highly energy-consuming process because the RNA polymerase must walk along DNA and making RNA as it goes along. In addition, any repair process in the wake of transcription, exactly as Anjana described, is again energy consuming because you need enzymes that come there, consume energy to sort of tie everything back together. So there's certainly no doubt that chromatin is itself, there are many experiments that point towards this at a biophysical level. They tell you that chromatin is at its core an extremely active structure. And we have to be able to account for this activity in some way. So let's take the following point of view, that in regions that are more active, where there is more consumption of energy, chromatin should see larger mechanical forces. There is more stuff being pulled and pushed because energy is being consumed. And in the whole process of remodeling or in transcription, we are distorting locally the DNA, and we're using energy derived from ATP hydrolysis in order to do that. The forces in different regions are unrelated to the other. So once you separate them large enough, again here, what is happening in this section of chromatin has nothing to do with what's happening in this section of chromatin. These are all independent, okay? So let's take these ideas together and figure out how to model them. So the first step is not to work at the truly molecular level, and that's where we make a specific modeling choice. We could start off at the truly base pair level, being completely molecular, but in the spirit of ignoring certain level of detail, in favor of assembling a description at a somewhat higher level, what I'm going to do is to describe a description in which you take each chromosome, the DNA inside each chromosome, and group it into levels of 1 megabase each, 1 megabase segment, and I will call this a monomer. Let this be chromosome number 1. I know what the sequence is. I can group it up into 1 MB units, and I know what the genes are on chromosome 1. So I can say that, look, there must be a different gene density in this monomer versus this monomer versus this monomer. The next step is to say that the activity that I must assign to each of these monomers must be set by the amount of transcription that is going on of the genes contained in that monomer. So right now we're making a statement that puts the biology back in. I have described my chromatin, or my DNA and the chromatin attached to it, and all of the other stuff attached to it, in terms of a polymer description of many monomers attached together. But now I say these monomers are not alike. And the reason they're not alike is because they contain different numbers of genes, and these genes have different levels of activity. So going back to the earlier idea, if it's more active, I say there are larger fluctuations. And to describe something that fluctuates more, the usual physicist trick is to say it's at a higher effective temperature. The effect of an increase of temperature is just to increase molecular motion. An increase of molecular motion is a larger fluctuation, and much of physics is really based on ideas that are as simple as this. So a higher effective temperature is to be associated with regions that are more active, and with less active regions, we'll associate a smaller fluctuations and a lower effective temperature. Let's now go a little bit into some of the physical ideas behind our justification for what we are doing. First of all, our active processes are emphasized are all happening in the nanoscale region, maybe about 10 to the 20 base pair regions. Each of these individual microscopic events is an energy-consuming event. It exerts forces. And that translates into these random forces on monomers, which we think of as being the analog of a temperature. So it's really thinking of this as though each monomer was seeing a different temperature, and that different temperature was correlated to the activity of that monomer, and that activity was correlated to the number of genes that were being actively transcribed in that particular segment of DNA. Okay. So the simplest model is just look at the gene density across each 1 MB segment on each chromosome. Because you know the human genome, you know exactly what this number is. Let's just set a cutoff arbitrarily. Let's take the top 5% of monomers by their gene density. Let's call them active monomers because they have a high gene density. Let's say that these active monomers, these top 5% of the total number of monomers, see a larger effective temperature. And let's choose a scale for that to be larger by a factor of 20 over the ones that are in regular thermal equilibrium, which we will set just for the sake of argument to T equal to 1, because the room temperature sets a unit of temperature over there. Okay, so you have hotter regions and cooler regions. Here's where the biology comes in. Because I know where the genes are, each chromosome presents a unique pattern or a fingerprint of active and passive monomers. 18, which is a gene per chromosome, if I depict my cutoff as that line, that red line that you see there, you can only see one monomer that sticks outside, sticks beyond. There's only one active monomer that chromosome 18 contains. Look at chromosome 19, which is gene rich. Chromosome 19 has 1, 2, 3, 4, 5, 6, 7, 8 monomers that sit above that background and therefore are active. So chromosome 19 is therefore a more active chromosome in the sense of having more active monomers than chromosome 18. And this reflects the underlying biology at the level of sequence and the level of activity. So then one does a complicated calculation. What we do is we simulate individual chromosomes within a nucleus. So these are a highlighted chromosome 18 and chromosome 19 and a background in a grayscale background of the other chromosomes. And from this, we can extract a sort of theoretical predictions that we can compare to the experimental data. First of all, if you asked, where are the genes? You know where the genes are because you know the sequence, you've mapped it onto this polymer model. Is there a distinction between gene rich and gene poor that is a spatial distinction? And the answer is yes. If you did this in a sort of cutout sphere representation, where the color represents the gene density, you can see more gene density towards the center, more genes to the center, and less gene density, fewer genes towards the outside where it gets darker. So that certainly works. The spatial separation of euchromatin and heterochromatin is something that comes out naturally from the model. We didn't have to do anything to that. Here's experimental data on chromosomes 18 and 19. As I said, 18 and 19 have approximately the same size, but they have very different gene densities. So chromosome 19, gene rich, is the sort of blue line on the bottom right. You can see the peak more towards the center, whereas chromosome 18, which is gene poor, is the one that is shifted out to the right. It has a peak towards the periphery of the nucleus. To compare that, chromosome 12 and 20, human chromosomes 12 and 20, have comparable gene density, even though they have very different lengths. Okay, gene density is total number of genes divided by total length. And you can see that these distributions are actually fairly, they look alike, whereas chromosome 18 and 19 have very different distributions. What does the theory predict in this case? So that's what the theory predicts. You can see that chromosome 18, you can see the lines that I've now drawn through those points are now my theoretical prediction. And you can see that it predicts that chromosome 18 has a peak which is much more central, whereas chromosome 19 has a peak which is much more central. Chromosome 18 has a peak which is much more peripheral. Whereas, as expected, for chromosome 12 and 20, I don't really expect to see much of a difference, and there isn't much of a difference in my data. What about territory? This is the experimental picture of the territories. You can see each of these colors separately. Each is colored a separate chromosome. And these are pictures that you get from the simulations. You can see that the nice separation of colors, the green, the blue, the orange, the red, the purple, etc. These are simulations that start from many different initial conditions. Each chromosome is a different color. And you can see that all of these final configurations incorporate territoriality in it. It follows very naturally from the model. So territoriality is another very robust prediction of this particular model. Finally, this is the center of mass distribution. So each of these points with their error bars tells you what is the mean position of the center of mass as a function of, so the y-axis is center of the nucleus to the periphery of the nucleus. The x-axis is chromosome size. So this is a very weak correlation with chromosome size that I showed you about earlier. With a model like this, I can do exactly the same calculation. I can look at the center of mass of each of my chromosomes 1, 2, 3, 4, right up to 22, 23. And I can ask, how does that look like if I plot it in the same representation? So that's what our predictions look like. You can see the same sort of qualitative behavior. And interestingly, you can see that the X chromosome is shifted upward, the H chromosome is below, even at the level of direct comparisons, and certainly within error bars, even at the level of direct comparisons with what is seen in the experiments, the theory actually does fairly well. Okay. So I would say, looking at that, even with my most skeptical physicist hat on, that predictions that agree well with data, even at a quantitative level. And they also agree with a broad understanding from lots of different measurements. Each of those was a separate measurement by a separate group. The group that did the center of mass location was not the group that did the locations of the distributions of chromosomes 18 and 19, was not the group that did the territory formation. So the fact that you have a theoretical framework that can address many different experiments is also very interesting. And any agreement between theory and experiment is fairly unusual in biophysics, especially biophysics of living systems. So the simplest model that I told you about had no cell type specificity. The number of genes averaged over a 1 MB region was a proxy for activity. And we used a simple model for how chromosomes interact with the shapes of chromosomes, which is called the random loop model, which is sufficient to generate compact chromosomes. But we could do much better because we understand much more of the biology. First of all, the first step is, instead of looking at just the number of genes, look at the transcriptome data and infer activity from the levels of expression. The second point is replace the random loop model by what is actually known about how chromosomes loops, by which you take high-C experimental data and look at the population average contacts of chromosomes and put that into the model. So that's a step in which you go from the simplest model, which actually works fairly well, to the more refined model, which takes in additional biological input that you imagine is important to tell you the difference between different cell types.
Gautam?
Yeah.
Can we take a natural break
Yep.
here? There are a few questions, and
Yep.
if you could
Happy.
re-go over the last part of the last slide, might be a good transition when we start again.
Yep.
Devashish, do you want to ask your questions?
I have two questions regarding the model. So, are the chromosomes assumed to be self-avoiding or ideal chains?
They're self-repelling. So they're not truly self-avoiding. They can cross. Of course, as you also know from Anjana's talk, there are enzymes that can do changes in topology. They can allow them to connect and cross each other. So it's a question of time scale. Given enough time, those processes will work. And we don't think that self-avoidance of chromosomes is going to be particularly important in this particular case. So they're self-repelling, but not truly self-avoiding. They can cross.
Local repulsion between the beads.
Yeah, exactly.
Then if I take two different chromosomes, they also self-repel each other in the same way.
Repel each other, exactly. Yeah.
So then this question that each of these beads now, many of these beads are being kept at different heat path.
Yeah.
So what happens there will be heat transport, right, along the chain between the chains.
This is a system that is not in equilibrium, but remember that these chromosomes are also diffusing around, so they're actually moving around. So that helps to even out there is a net effective temperature gradient between the center of the nucleus and the outer part of the nucleus, because the activity also shows a gradient. It's more active towards the center and less active towards the outside. I'll show you some examples of that. But this is not a problem in terms of thinking about steady states. This is a steady state. This is an effective temperature, not a real temperature. So I don't have to worry about real temperature issues about thermal gradients in this particular case.
Just one point I wanted to clarify. Is it like, if I think about them as if like each of the bead is carrying its own heat bath along with it? Is that right?
That's perfectly right. That's perfectly correct.
Okay. And then the nuclear membrane, that confinement is modeled by local hardcore repulsion.
Yeah, it's just a hardcore repulsion. Yeah.
Okay, thank you.
It's completely confined in that. We wanted to look at softer nuclei when we were thinking about since modeling stem cell nuclei. We haven't done that yet. It actually turned out to be very hard to move away from the sphere, and the student who did this did quite an amazing job of even looking at more flattened nuclei, but that's a very hard calculation to do.
Thank you.
Vijay?
Yeah. Gautam, does the territoriality disappear if you have an equilibrium simulation?
The territoriality doesn't completely disappear because that depends upon how how compact you define your chromosome to be. If your chromosome is highly sort of internally connected and is a pretty much compact object, then it doesn't tend to overlap much. But what you lose immediately is any distinction between chromosome 18, 19, 12, 20, etc. They just put it at random. So you need to be able to have both of this information. What makes chromosome 18 sit in one location, chromosome 19 sit in another location, as well as the fact that they form territories.
Okay. So two follow-up questions then. So one is, suppose I actually did a simulation, an effective simulation where I had a temperature gradient from the center of the nucleus to the outside, something like a thermophoretic effect.
Yeah.
Would that be enough?
That wouldn't be enough. I mean, that would not select one over the other. If at all, there might be some complicated entropic effect that comes from the fact that they're not all the same size. And so maybe the smaller ones might sit towards the center, the larger ones might sit towards the outside in the presence of an externally imposed gradient. We haven't looked at that.
Exactly.
That's the only possibility that I can think of.
Right. And also, I mean, in the model, I was a bit surprised. I mean, you have so many, how did you get so good fit between the model? And you chose some t equal to 20.
So I'll tell you about the little more complicated model later. And it turns out that the model is pretty much, and the results are pretty much insensitive to that, to the factor of 20. As long as there is some difference between inactive and active, that's enough. You can push it down to a factor of 4 or 1:4 or 1:6 or something and get equally good results. We chose 20 because the hydrolysis of ATP is typically corresponds to about 20 times T is what we, but it isn't necessary. I'll tell you about a more detailed model as we go along.
Sure.
Pradeep?
Yeah, Gautam, I was wondering, how does this coarse graining size you took as 1 MB monomers, right?
Yeah.
So about 50 or 60 of them.
Yeah.
So how, if you make it smaller, I am sure they will still separate nicely. If you make it bigger, beyond a point, I suppose you will lose this separation of, shall we say, 18 and 19. Right?
Yeah, we chose 1 MB because the general thinking in the field is that is a building block for territories, that you don't need to go much smaller than that, and you should not go much larger than that. So that was a choice. To address some specific questions with Hi-C, we tried going down by a factor of about 10 to about 100 kb. And things sort of work, but it numerically gets complicated and sort of much longer to simulate. So the 1 MB is about just in the right ballpark for you to get sensible answers, good predictions, and to be computationally tractable.
Yeah, but within that 1 MB size, there can be genes which are active and genes which are inactive, of course.
Yeah, not genes, monomers. So totally, this total simulation has about 6,000 odd, 6,400 odd monomers.
Yeah.
And some fraction of them can be active, some of them can be inactive.
Yeah. So, but isn't it almost surprising that this works so well, given there are these specific topologically associated domains, and you will have anchorings at this point, and this part will be transcriptionally active, and then very sharp cutoff. And
so let me go on. Some of these questions I'll address a little later, but that is an important point. I mean, it looks so good. Why does it have any right to be so good? So I would argue that I think that the basic intuition is correct, and it's good. I'm surprised myself at how well it does.
Yes.
But I would think that the idea really is that the intuition behind it is the right intuition, that what we chose to keep and what we chose to throw out is in fact what dominates.
Okay, Gautam, I will ask a very simple question and then maybe we should restart. So if you look at human cells, at least, one of the X chromosomes gets inactivated. So does the position of the X chromosome depend on whether it's inactivated or not, being peripheral versus outside?
You're stealing my thunder. That's a high point of the talk. I'm going to get to that now.
Oh, I see. I'm so sorry. Well, the floor is again all yours.
Okay, so how do we put in cell type dependence upon this? So as I said, one way to do it is to go back to transcriptome data, use RNA-seq. So this we took from the ENCODE collaboration. You average expression over 1 MB ranges. And then what you see in that little figure there is a plot of the log of the expression on the y-axis. And this is sort of monomers in order of increasing expression on the x-axis. So you can see that for this particular cell type, the GM12878 cell type, as well as for all cell types, it has a very characteristic shape. It rises fairly steeply, then it sort of shows a plateau, then it goes up again. So one natural way of thinking about it is to say that, look, everything in the sort of sharply rising up point is going to be pretty much in thermal equilibrium because it's a log scale on the y-axis, okay? The point where it saturates, we can say, let's just fix that as some sort of floor of activity. Let's call it about, say, 4 times room temperature or 6 times room temperature. And the path that goes up very steeply, we can say these are the far more active monomers with lots of genes inside them. And that we have to worry about how we assign activity to that. Okay. So we did a lot of study of this, what it looked like, what might be sensible in temperature assignments, what are these limits, et cetera, et cetera. And we finally came up with a model that we thought had all of the right ingredients to do a good job on this. For this particular cell type, there are at t equal to 1, that is the increasing part of the expression, there are about 48% of monomers are just there in that limit. At t equal to 6, which is the flat plateau part, more or less plateau part, we have about 40.2% monomers. And in the sharply increasing part is about 8 to 10 monomers. And the maximum temperature here is 12. It doesn't matter too much, as I said, how you actually divide this, but we're going by the shape of the curve of the expression versus the monomer in increasing order. And as I'll show you a little later, this is what it looks like for all of the different cell types that we looked at. The 12878 HMEC, HUVEC, IMR90, NHEK, etc. And that's all of them plotted together in the top left figure, where it says all. You can see all of them have a sort of common shape. Okay. It's important to specify that a monomer in IMR90 could be inactive in IMR90 but active in HMEC. So it's only the overall shape that is conserved and not the individual activity or inactivity of a particular monomer, because that's what makes these different cell types different from each other. The activity, the expression, the log of expression versus monomer picture on the left-hand side translates into an effective temperature versus monomer on the right-hand side. That's a rule that we use to assign effective temperatures to monomers for all of these different cell types. And on chromosome 12, for example, the 2 copies of chromosome 12 are shown for each of these cell types, sort of brought towards the center. They're kind of slightly different. The numbers of monomers that are active in the NHEK differs from the number that are active in the 12878, etc. So there are questions of detail in this, but certainly this is one way of incorporating cell type dependence of activity in each of these different cases. Once we do that, we can throw the whole kitchen sink at this, generate the distribution for center of mass, the contact numbers, and the shapes of these. And there's, we can look at the 2-dimensional projections, we can look at the roughness of chromosome territories. None of this has ever been attempted in any simulation before. So we had a field day with doing all of this stuff for the first time. And all of this is really the very hard work of an outstanding graduate student of mine, Ankit Agarwal. And so we were able to do a whole lot of this. Let me now go take you through the results here. So the picture to the top is a sort of cutaway description from the simulation, which shows the dark regions there are heterochromatin, the light regions are euchromatin. So you can see that the outer band, the periphery, is heterochromatin, and inside the lighter regions are euchromatin. You can look at the middle picture, which shows you the gradient of activity. The effective temperature is largest at the center and smallest towards the outside. That has a parallel in gene density, which is largest towards the center and lowest towards the outside. So you can see that averaging over many configurations of these in steady state generate the same sort of generic features that you see when you think about euchromatin, heterochromatin differences, gradients in gene density, etc. Here's actual comparisons, again, cell type. There's only data that we had for the GM12878 cell type. So you can see the distinction between 18 and 19, 12 and 20, center of mass distribution. Kind of reasonable. It's a little easier to see differences in the center of mass distribution than for the DNA density distributions as a general sort of feature. But of course, you have to average much more to get good data for the center of mass. Because we can look at each chromosome independently, we can construct territories for each of these chromosomes. So those are the pictures that you see on the top left. Chromosome 1, 2, 3, 4, 5, 6 with their copies. And then you can look at how rough is it? What is the nature of the ellipsoid that I must construct to enclose all configurations of this chromosome completely? What is the roughness of the structure that I will get? What is the ellipticity? How far away from a circle is it actually? If I project it onto 2 dimensions, that's a technique called 2D fish. What does 2D fish behavior look like? And then I can ask all of these things, okay? Look at the picture on the bottom right, the figure on the bottom right. So these are 2 3-dimensional parameters. One is called regularity, one is called ellipticity. And that's a comparison between the GM12878 data, the predictions from the simulation for the GM12878 cell type, for the IMR90 cell type, and the experimental data on a cell type that is not exactly, but it's closely related to the IMR90 cell type. You can see that the qualitative behavior is the same. In fact, the sort of rise, that flip, and then sort of going up that you can see for 22, 20, 16, 17, 11, 17, 19 towards the right of the figure is kind of consistent pretty much. I mean, this is a question of detail, and even between cell types, you can see a fair amount of variation. But overall, I'm sort of enthused by this reasonable agreement with experimental data. If you look at where the active monomers are vis-a-vis the inactive monomers, if you just plot distribution functions for active and inactive monomers and pick chromosomes 1, 13, 19, and 21, you can see that the active monomers tend to be a little more towards the inside than the inactive monomers. And again, this has been seen, sort of the fact that active alleles are positioned more towards the interior of the nucleus has been known for a while. Now we come to this sort of perennial question: are chromosomes distributed according to their gene density or according to their size? And to what extent do these distributions vary across different types of cells? Okay. So here is a chromosome size correlation in the more complicated calculation for GM12878. So it's a kind of mere correlation. It's sort of more or less consistent with a gentle rise and a saturation, much as you see in the data, but the error bars are large. Okay. Look at it now for the chromosome gene density. And this is gene density per megabase in the x-axis and relative radial position on the y-axis. And you can see that both experiment and simulation are kind of more consistent with this type of behavior. You can see that chromosomes 17 and 19 are pretty much bang on the location between experiment and simulation. But the other points are also kind of consistent with this type of behavior. You can look at it across different experiments done on the same GM12878 cell type, FISH, Insitu, TCC, etc., and look at the correlation between the positioning according to chromosome size in the top row and gene density on the bottom row. And you can see that overall, it looks as though the fits are somewhat better towards the gene density. But as I said, data are noisy, and there's not much that you can do about it. But certainly experiment and theory are in the same range of error bars. So our answer to this question, are they distributed by the gene density or by size? Overall, we see that distributions by gene density provide a better picture for the more spherical nuclei. But if your range is restricted enough, you're only looking at a smaller number of them within the error bars, maybe you could think that, look, your size distributions are also okay. This is sensitive to the activity distribution and the choices that you make in the modeling. Okay, so we made a specific choice. These are the answers that we get, but we have changed this substantially. Promoted a lot of inactive monomers to becoming active or the other way around. Certainly these distributions and the structures of the distribution would change. Something that I think is important in the interpretation of the experimental data is the role of nuclear shape. Much of these experiments are done on somewhat flattened nuclei, and whereas all of the theory, all of the simulations are done on spherical nuclei. And so mapping one to the other becomes somewhat more difficult. And it's really my feeling that much of the interpretation, of the differences in interpretation, might really come from the fact that the nuclear shape is not accounted for when you move from the theoretical model, the simulation model, to the experimental model. We've looked at fractional volume occupied by each chromosome as a function of length, etc. Again, that's experimental data, and that's prediction for two IMR90 and GM12878 data, kind of consistent with the increase. Let me come to what I promised Sandhya, and that is this question of the distribution of the active and inactive X chromosome. So remember that women have two X chromosomes. One has to be inactivated, so there is an inactive and inactive. The inactivated chromosome tends to be much more tightly held together, more heterochromatic. It's called heterochromatic bar body. And the fact that these are located differently has been noticed at least for the past, I guess, 50, 60 years in terms of locations. So the fact that X chromosomes in female cell nuclei, both X's, are asymmetrically distributed has been known. Remarkably, this is also a feature of the simulation data. If you look at the distribution function for the X active and the X inactive peaks towards the outside, the X active peaks towards the inside. You can see that it's not a very large, substantial change as you can see for chromosomes 18 and 19, but nevertheless, it is very clearly there, as you can see from this particular image. And as far as I know, this is the first calculation ever to try and understand the biophysical underpinnings of this particular phenomenon. We can do things that I'm a little less happy with. You can look at contact probabilities. Of course, the way we structure it is that much of these contact probabilities are derived themselves from high-C data. So there's already part of the calculation. So the question of what to compare to becomes a little trickier. But we can look at contact property in cis and in trans across chromosomes, etc. I would not assign much importance to the actual contact map themselves because, as I said, there's a lot of information that goes into that already. But I would like to look at the picture that you see on the top left, which is the probability distribution of the end-to-end distance. The log of P of S is a function of distance along the probability that two monomers separated by an internal distance S are in contact is something that seems to decay with a power that is very close to 1 in the GM12878 data. This is pretty much bang on. The fact that P of S goes as 1 over S to the power 1.06, 1.08 is precisely what was seen and has consistently been seen in the data. We point out in the paper that this is not generic. It sort of varies hugely if you go across different cell types. It's valid over a fairly short range. And in fact, I think much of these hugely cited papers that said this is something very special about chromosomes, etc., are probably completely wrong because the region in which that scaling behavior is seen has shifted and become smaller and smaller and smaller. But I think that this is again one important feature of the simulations that is again borne out by experiments with all of the appropriate caveats. So let me summarize. Where am I? And yeah, 5:35 is a good time to stop. We talked about chromosome territory. I talked about the heterochromatin, euchromatin spatial separation. I talked about deriving the positional distribution of individual chromosomes. Within each chromosome, the distribution of active and inactive segments, the center mass distribution of individual chromosomes. I talked about the differential location of the active and inactive X chromosomes, the perennial question of are you distributing chromosomes by the center of mass or by gene density, and questions that had to deal in detail with the nature of the shapes and physical properties of chromosome territories, ellipticity, regularity, and a whole bunch of other parameters that we looked at. But let me just emphasize this particular point. All of these results follow once you specify the pattern of activity on each chromosome and how chromosomes actually loop. Given that, what is remarkable is that you're able to provide predictions that resemble those seen in the experiments across a whole bunch of different experiments. It's not very easy, but it's usually much easier to fit one experimental data or at most two. But the fact that you can fit multiple pieces of data from multiple experimenters and multiple experimental groups and extract essential features, if not exactly the right, the question of what is the right answer, of course, is a big question mark. But certainly the right qualitative features and behavior is, I think, somewhat unusual. So this model, I think of it as a first principles model in the sense that none of the stuff that comes out of it is put in by hand. This is just the way, this is emergent behavior that comes from the model. There are many contexts in which people have talked about phase separation in cellular context, and you probably had some talks about this altogether. But the phase segregation that I'm talking about in this context between active and inactive components is one that takes transcriptionally active components and brings them towards the center, effectively compartmentalizing them. So it's a sort of compartmentalization that is like the compartmentalization that is envisaged in phase separation scenarios, except that this happens completely due to activity. Okay, so it's interesting to try and examine the relationship between compartmentalization and these different ways of thinking about it. None of these results, not one, would have followed from assuming that chromosomes are polymers in thermal equilibrium. And which is why, because the community has very, very largely focused on thinking of chromatin in terms of an equilibrium description, that none of these have ever really been attempted even as potential calculations so far. So none of these experimental results have that this calculation has confronted has ever been reproduced by anyone else. There's a whole bunch of interesting stuff. There's the nucleolus, there's lamins. We didn't have the nucleolus in explicitly, we're going to do that. There's the role of lamins is extremely important. The role of nuclear shapes, very complicated calculation, but nevertheless possible. Making contacts more dynamic is something that might give us a better idea into how to relate this better to the high-C data. all of the stuff that DNA damage can do is something that's very interesting, because DNA damage, as I said, requires repair. Repair is an energy-consuming process. This leads to changes that are reversible in the positions of the chromosomes. Serum deprivation does interesting things that we don't understand. What happens to distributions in the context of development and in stem cells is something that we don't understand from a theoretical point of view. But the main bottom line is that I want to make is that inhomogeneous activity, confinement, and chromosome contact really dictate large-scale nuclear architecture. And this is different from the sort of approach that Anjana had described, where those questions are questions that are detailed biochemical local questions. But the question that I think that physics can answer, or physics can attempt to answer, are all questions that happen at a much larger scale, the 1 MB and beyond. And they have to do with centers of mass of chromosomes, where are they located, the shapes of chromosome territories. These are not very natural questions for purely biological approaches, because biological approaches don't really, it's sort of an interesting question of, are these relevant at all at any level to function? Certainly the locations of chromosomes and how they move under serum deprivation, etc., is certainly relevant to function. But are some of these questions, the distribution functions themselves, are they important, are they not important, is an interesting question that I really would like more insight into. I don't have that at present. Here's a quick view of something that Tejal has been doing, the postdoc with me at Ashoka. We've given sort of quarantine requirements. We haven't actually interacted well enough. This is something that she started together with my old student, Ankit. And this is an example of how the nucleolus forms. So we've sort of, the nucleolus is sort of as a paradigm for phase separation. So if you look at nucleolar organizing regions on the different actocentric chromosomes, 13, 14, 15, 20, 21, 21, 22, you can see how they form, how these are initially separated, but then they seem to come together in this particular form. So how do you combine this with the sort of calculation that we've been doing is something that we're going to look at in the future. So all of this work is done with really remarkable people. Nirmalendu Ganai, who really did the initial calculation. Shuriji Sengupta, long-term collaborator of mine, and an absolute gem when it comes to thinking about anything quantitative in terms of computation. And my outstanding student, Ankit Agarwal, currently at Weizmann, moving to Germany, who put many of these calculations together and really did an amazing job. The most detailed of these papers is the Biophysical Journal paper, so I can recommend that you read that. And my quick summary of chromatin as an active polymeric material is one that was circulated to you little before this. Okay, so that's all I had to say. Thank you very much, and I'm happy to answer as many questions as you guys might have.
Thank you, Gautam. That was interesting and actually very exciting to hear. There are several questions. Sonali, do you want to unmute yourself and ask?
Yes. Thank you for that really insightful talk. So I'm assuming from this talk that activity correlates with organization and architecture, but then at the same time, activity is sequence information dependent. So is it a general principle or rule that lower level sequence information determines higher level organization, or organization emerges from sequence information in eukaryotes, but this organization is not so in bacteria? Your comments, please.
Very good question. Yeah. So bacteria tend to have fairly prolific, levels of transcription. I think probably 98% plus of the bacterial genome is transcribed at some level or the other. Whereas here, it's important that you have regions that are not transcribed at all. The substantial fractions of the genome are not transcribed in a sort of active sense. Whereas some parts that are especially related to housekeeping functions are highly transcribed. So that you do need to have this sort of small part of the genome that is highly transcribed, large part that is not transcribed, to get the sort of structure that I told you about. So I would imagine that there is nothing interesting that this type of model can say for bacteria. It's really for higher eukaryotes that one can make predictions that are concrete in terms of nuclear architecture at the scale that I described. And the reason derives from this, that you do need this you need to have small things that are highly active and a large number that is just background and just along for the ride. But you're right about the first part, that this really is saying that sequence at the small scale leads to structuring at the large scale through the sort of indirect route that involves activity. And that is a good point. That really is also at the center of what I'm saying, that one can make cell type specific descriptions and put them into the model and get some useful information. Overall, the overarching sort of result is that there's not very strong dependence upon cell type. But there are, at a subtler level, there are. Whether experiments can see these, or whether we're capturing them correctly, or whether the effects of the lamins have to be introduced at various points, and what does the nucleolus do? We put that in. All of these we are yet to investigate.
Thank you.
Mohinish?
Hi, thank you for a really exciting talk. I had a quick question about sort of within chromosome inhomogeneity of active monomers. So, I don't know what the assumptions are, whether they're kind of randomly scattered across the entire length, or whether you see whether they're located in the middle versus the edges, if that makes a difference to the overall positioning. And I was also wondering that if you add in sort of the total length plus overall activity plus any inhomogeneity, then does the sort of the composite model fare better in its how it relates to the overall positioning as observed in the experimental data? Because you showed individually the length, individually the activity, but if you combine them, does it improve?
That's a good question. So if I fit it to some combination of length and gene density, would I have a better fit? I don't know. That's a good question. And ideally, what I would like is an experiment that is able to do all this on the same system. Right now, we're looking at experiments done by different people under presumably not exactly the same conditions. And then it becomes a little difficult to add this interpretation to that. And my suspicion is that there probably is a sensible way of combining both length and gene density information, at least from the theoretical point of view. Whether there's something experimental to compare to, I don't know, but that's a good question. The second point is that the earlier question was that we don't actually do anything. We take sequence in, we take activity, we take transcription levels, gene expression levels in directly, and then we translate that into our model. So we'd have no freedom in determining which monomer is active or not, whether it's a center, whether it's peripheral. Anything where we know that where the activity exceeds a certain level because the genes that it contains, we say that that is active. And we know exactly what sequence on what chromosome it maps to. So we have no flexibility there at all.
I see. And if I can just add a quick comment that it seems that, just from a purely physical perspective, if you have active genes and you're transcribing, you want these molecules to exit the nucleus. So from an exit perspective, you'd want them to be to the periphery rather than the center. So do you think it's some kind of a fight between biology and physics, where physics is forcing them to the center for purely mechanical reasons, when really, ideally, biology would like them to the periphery for easier transport out?
No, I mean, I'm always very skeptical of these just so stories, because you can always concoct a story that fits with your particular way of thinking about it. So one approach to why you have euchromatin towards the inside and heterochromatin towards the outside is called the bodyguard hypothesis, that the heterochromatin, being less useful, acts as a bodyguard to prevent external insults to the euchromatin, which is useful and gene rich. So that's a perfectly reasonable just so story. But again, it's that they don't really give you more insight into why something happens, why it is maintained versus why it's not. And what is being optimized is usually a very hard call to make. So because ATP is small, highly labile, highly mobile, etc., I would think that the diffusion time to the periphery is probably not an important consideration over the time scale that we're thinking about. So that may not be what biology is trying to optimize in this case. That would be my guess. I could be wrong.
Thank you.
Pratyush, do you want to unmute yourself?
Yeah, actually my question kind of got covered. My only comment was that exactly that if you in your experiments also had this idea of which cell type and in which condition your cells are, then I think this model would be much more, the error would really reduce and help you. Is that true?
So we can do cell types. I showed you 5 cell types, and there are subtle differences between them, but overall they kind of look the same. And I think that's also consistent with what one generally knows. Now the question is, can you think of conditions that are sufficiently different where you can see, for example, 18 and 19 chromosomes completely interchange their position? And can you ask what is the effect of that change in condition on transcription programs? If we have that information, we can put it to the model and see, does the model reproduce that? And certainly the model is able to reproduce non-trivial distributions of these. You can, by just hiking up chromosome 12's activity, I can push chromosome 12 in or outside. So if I know that that is a response to a particular condition, I can put that into the model and ask, does the model predict that or not? Again, the error bars are large.
Yes.
The amount of averaging that you need to do are large. So so whether a model is right or wrong is a sort of harder issue to, or better or worse is kind of much harder to settle. And at this stage, I would settle for more or less the broad features that I discussed.
Oh.
The heterochromatin euchromatin separation, the gene density, the roughness of chromosome territory. Right. There are a couple of these features saying that, look, we can calculate these within a consistent model. And this consistent model has prediction that resemble those that are there in biology. And to do a much better job, I would ideally like much better data consistently by one group on one system.
Yes.
So there can be much more control on that.
Yes, yes. Because because from my understanding, I think this is seen that different cell types or cells from different origins or tissues have different locations of specific chromosomes, and that could affect your data slightly.
Some of the data I have seen has not been, there is large amount of noise in the data. There's not enough statistics to make very strong points. And I suspect that some of that is due to a feature that we haven't included, which is the interaction with the lamins. So that will favor certain chromosomes attaching to lamin-associated domains to the periphery. And so that can also change. Remember, we're always talking about small confined systems. And whenever you have a small confined system, the interaction with any boundary becomes important. So that will help you shift these locations a little. This is something that we tried out earlier. We haven't explored enough, and hopefully we will be able to do a little bit more of that in the coming year. To ask, can we put in that level of one further layer of control through the lamins and lamin-associated domain, and ask, can we see differences in cell types that are accentuated in such a situation?
Okay, thank you.
Debashish, his hand has been up for a while.
Gautam, fantastic talk. One question I had regarding the contact map and contact probability distribution. So did you use the contact map to model the polymeric structure of given DNA, given chromosome?
Yes, we did. We did that for all the chromosomes. So we took the Hi-C data, and then we thresholded it. We looked at the strongest contacts. We said that we averaged that over the 1 MB region that we were considering. So if you had contacts that survived the strongest contacts between these things, you attach those two monomers with a permanent link. Okay, so you are putting that structure directly from Hi-C. Of course, the problem is that Hi-C is an ensemble measurement. So it's the single cell to single cell variation could be much larger than what you see in the ensemble. We don't know about that yet.
So what I'm trying to understand is that you have from Hi-C data, you are taking the structure, and from the gene density, you're taking the activity.
No, the activity is taken from actual RNA-seq data.
Okay, and they are localized according to the contour-wise on the effective polymer. They are localized in space. According to the experiment. Okay, associated with this, I had this question about the if your chromosome is generated by using the Hi-C map, then doesn't it automatically constrain your contact probability distribution?
It does, but there are not too many contacts, because once you sort of threshold the level at which you allow two things to contact each other, it turns out that the number of, we call them loops because they're sort of permanent loops because of permanent attachment, that turns out to be fairly small. I think totally of about 6,400 monomers, there are only about 100 to 200 loops is what I remember.
But in the contact distribution, this is actually special. If the special separation between two monomers becomes smaller than certain cutoff radius, that's part of.
Yeah. So I'm saying that the P of S as well as the contact distribution, they take information that comes from the Hi-C maps, but it's information that is thresholded, that is incorporated in a particular way through permanent bonds, and then put into the data. And then you ask what is the effect of that on the other parts that you have not allowed to be directly in contact.
Okay.
So that I'm not too happy with the Hi-C representation, but it's at some level it's the best that we can do. And interestingly, the purely random loop model does very well. sort of, people talk about high C and the importance of high C and a whole bunch of this, but at least I think you can get equivalent results just by random loops.
Oh, okay.
That's been pointed out in other contexts as well.
Samarth, do you want to unmute yourself and ask it?
Thank you so much for the talk, sir. I wanted to know, is the relative position or location of the chromosome territories important? Or does the territorial arrangement that emerges in the simulation match with that of the experiment?
It does, because I showed you the distributions, no? I mean, so it's not as though that they have any, it's all statistics. There's a sort of distribution of possible locations of the center of mass or the DNA associated with that, and that matches. That's what I showed you. So chromosome 19, which is gene rich, is always found more interior. Chromosome 18, which is gene poor, is always found more towards the outside. And so we looked at I showed you the distributions of each of these, of the center of masses for each of those chromosomes compared to the experimental data. So all of those are largely consistent. And also we compared it to as a function of chromosome size as well as chromosome as gene density. All of those are consistent with what is seen in experiment.
Okay, thank you.
I'll ask Muskan's question, which he or she wants me to. Can we comment on the positioning of the nucleolus based on your model? As per my knowledge, nucleolus is somewhat peripherally placed, like close to the nuclear envelope, but highly involved in ribosome biogenesis.
Great question. So in this calculation, we didn't do the nucleolus, and in fact, no other model of higher order nuclear architecture has included nucleolus so far, as I know. Certainly up to last year. It's very much on our mind. And the last picture that I showed you was Teja's pictures of the nucleolus simulation. I would expect that because those are highly repetitive regions, they have activity that derives from different reasons. So how to put an activity in the nucleolus is a little more complicated, and I have not thought enough about that. So I can't answer right now the question of will that be more peripheral, will that be more central? But it's very high on our mind to look at that. Our initial question was, can we show the same sort of phase separation behavior that has been seen in the experiments where you have these nuclear NORs coming together and forming. And after that, and then the question of what does that mean for the distribution function? After that, we'll ask exactly the sort of question that you had in mind. Is there a preference? If I look at the central mass of the nucleolus, and once it's sort of settled down, where can I find it? Is it more outside? What can I look at that distribution? So great question.
Gautam, related to that, you said that the activity in the nucleolus is of a different kind. What do you mean by that?
No, I think the nucleolus forms in the cell cycle, disassembles at mitosis and forms in G1, and then there's fusion, like as
yes,
phase separated domains would do. But apart from that, the phase separation of the nucleolus is driven by this intrinsically disordered nucleolar proteins and also by rRNA transcription, right? Both of them drive this phase separation. Now, both of these can be affected by conditions of stress because the nucleolus, meaning rRNA production, of course, will go down under conditions of stress. So does one expect the nucleolus also will fall apart under conditions of stress? And I was wondering, eventually put one
we can do this. This we can certainly do. I've been interested more in the question of how do these things get together? How do they phase separate? And can we model that in some simple polymeric way? And then ask what does activity do on that? But we can also ask this question: if I high cut the activity, if I change the activity in some form, do they tend to break up? And if they tend to break up, how do they break up? And one question would be, is this 1 MB description sufficient, or should I be more microscopic? So what in the current simulation, we sort of look at the regions that form the nucleolus at a 100 kb level. So that's 1 order of magnitude smaller than and the rest of it we teach at 1 MB. So we have to make certain modeling choices there, but they kind of seem to be the right ones. But this is a question that I can hopefully answer in another 6 to 8 months.
Yeah, but the NDO model would be agnostic to this individual nucleolar proteins which are supposed to drive this separation or rRNA transcription.
Yeah.
I think there are a couple more questions, but I'm going to encourage those people to put it in the Slack channel, so Gautam can answer it, including Pratyush, your second question. I know nothing about chromosomes and DNA. I know it shouldn't be like that since I'm a biologist, but I really genuinely don't know much. And thank you, Gautam. It was very, very useful and really very exciting. And thank you all.
Thanks to all of you. Thanks to the organizers.